This presentation is on the biology IA, and I'll be going through what it is, how you would choose a topic, the general criteria, sort of an approach of how you can approach this project in a step-by-step manner, some tips and tricks, and some questions at the end. So first of all, what is the biology IA? So you probably already know this, but in general, or in the past, it's accounted for 20% of the final assessment, and it's a single individual investigation. So you have to do it on your own. You can't share data with anyone else or do an experiment with anyone else. It might involve a hands-on approach, the use of databases. You can use modeling, simulation, or a hybrid of these things. And it's internally assessed by a teacher and externally moderated by the IB, which this might also be changed because of the pandemic. So in terms of choosing a topic, it can be challenging to come up with an original topic as they ask you to do, because of course there's already so much out there that's been done. But a really good place to start is just with what topic interests you. And to investigate something you don't already know the answer to. And this is to say, basically, try not to use a too common procedure that you can already find on the internet. So if the procedure is written in full, the way that you've written it in your IA already online, then you have a pretty good idea that you probably want to modify that so that you can demonstrate more initiative and more creativity and just generally more engagement with your IA. And then if possible, it would be good if you could use primary data, and you need to have both quantitative and qualitative data. Then also when choosing a topic, it's important to consider ethics in the environment. So for example, using animals might not be the best idea, even though it's, of course, biology, and the environment. So if you're using a lot of chemicals or things that are environmentally harmful, you might also want to rethink this topic that you've chosen. Then here's a general overview of the criteria. You can look back on this and it's present everywhere. I'm going to go through them step by step. So first of all, criterion A is personal engagement. And this one essentially has to do with showing independent thinking, creativity, or initiative. This is sort of what I was saying with your choice of topic and your procedure. So if this is how you can show it throughout your IA. It's a holistic criteria. You don't just want to write a short paragraph at the beginning with some kind of anecdote about how you're lactose intolerant and that's why you're studying lactose or something like that. You really want to incorporate it throughout your entire IA and continuously justify why you're interested in this through your methodology, your implementation, the presentation of the investigation, or something in that vein. Then secondly is criterion B, which is exploration, where you'll want to include the topic of your investigation and have that clearly identified, and then the research question, which is a component of that, the continuation of that, and have that be relevant and fully focused, which is to say you need to include all of your variables in that. And essentially, whoever's reading your IA should, from your research question, essentially already know what this IA is going to be about. Then you should also have background information, which needs to be appropriate and relevant. So don't just write about the entire topic in the syllabus, but really just things that will enhance understanding of the context. And if you need a little bit of additional information that might not be in the IB syllabus, you can include that here just so that there's a greater understanding. Then for the methodology, this basically really has to address your research question and make sure that you're taking into consideration any factors that could influence the relevance of your experimentation. So if anything might influence the validity of your investigation, you need to make sure that you're accounting for that in your methodology. And essentially, whoever is reading this needs to be able to do your entire experiment just from the methodology without asking you any questions. So it needs to be complete. Then safety, ethical, and environmental issues, as I said, need to be considered, and it's really important to just have a signposted section on this where you demonstrate that you have considered these factors and you're considering environmental, ethical, and safety issues in this experiment. Even if there aren't any, for example, ethical issues, state that there aren't any, just so that you've covered your bases essentially. Then for criterion C analysis, in terms of raw data, there should be sufficient qualitative and quantitative. So you need to have enough trials, at least 3, probably more, and also have quantitative, so observational things like color and stuff, and qualitative or quantitative as in numerical data, basically. Then data processing should be appropriate to the raw data that you have and the investigation, and should be accurate enough to come up with a conclusion that's consistent with the data. In terms of uncertainties, you should consider these and write them basically like in your methodology if there's any equipment uncertainties or any other uncertainties, and also consider the impact of these. Then in terms of your interpretation of the process data, there should be correct interpretation, which is just to say that you're interpreting what is already present in your data. It doesn't have to be necessarily, well, I mean, it should be, but if it's not in correspondence with the sort of general accepted scientific context, that's not as big of a problem as if it's just not relevant to your data. Then, in the evaluation, you'd have a conclusion, which should be your conclusion from all of your data. So what you've gathered from all of this should be described and justified. It should be relevant and relevant to the research question, once again, and supported by the data. And you should compare it to the accepted scientific context. Like I said, if it's not like the scientific context, like if you reach a conclusion that's totally different from the scientific context, just say that. Say that the scientific context is this, and I've come to this conclusion, and they're different. And try to, of course, justify that. But if it's not the same, then that's not the end of the world. Then you need to look at your strengths and weaknesses. So limitations of data and sources of error need to be discussed. And for those things, you should also mention improvements. So realistic and relevant suggestions for how you can improve the things that you have previously done wrong or could have done better. And then also what's good to include is an extension of the investigation. So how would you take this further? What other experiments would you do? Just like how would you continue this experiment and this investigation if you could? And then lastly, communication. This is another holistic criteria, so it's assessed throughout your IA. And basically it's about how well structured and clear your presentation is. So the necessary information needs to be present and presented coherently. This is really important because the marker that's marking your IA is not going to spend a lot of time on it. So the more clearly you've presented your information and the easier it is to look at and understand, the better you're going to get marked, essentially, not just for this criterion. Then it should be relevant and concise. And concise, just try not to waffle too much. And you're not going to be able to fit it within the page limit anyway if you're not concise, but it also will not aid your conclusion. Then use subject-specific terminology and conventions. Don't use lay terminology, use what you've been taught in class. It's really important to be able to apply your scientific knowledge that you've gained from taking IB biology, which is basically what they're testing here. And then in terms of errors, it's not that you can't have any mistakes, but they shouldn't hamper the understanding of the focus, process, and outcomes. Then, in terms of the approach, firstly, you need to identify, this is just like a general timeline for you. It's very crude, but it might give you a step-by-step understanding of how to continue. So first of all, try to identify a topic that interests you, or like if you're not that interested in any of them, one that you wouldn't mind spending more time on or learning more about. And then do some research. It's also important to talk to your teacher and discuss what resources you have available to you. So you can't do, if you don't have like an incubator present and you need one, then it wouldn't be helpful to write a whole methodology that requires an incubator. So just talk to your teacher about what you have available to you, and then narrow down to research question. Then you would design a method, you would carry out the experiment and collect your data, and finally write the report. So in terms of tips and tricks, these are not really like sorry, we'll go back. These are just sort of my tips and tricks that help me, and also they're general ones from the IBs. So first of all, choose a topic that interests you, as I've said. Signposting and having a clear structure will really help the marker be able to give you those points and really tick those marks off for you so that you can get the points. What I think is a really good way to do that, for example, is putting things into a table format. So for example, for all your variables, like your dependent variable, independent variable, your control variables especially, is to note the type of variable in a table, give a description of that variable, and then a justification for why it's important. For example, controlling temperature, like what effect would that have if you didn't control temperature? Then include any uncertainties in measurement, and don't forget error bars in your graphs. Label tables and equations appropriately. Again, work with your teacher and know your resources, and present your process data in different ways. So for example, with graphs, with statistical analyses, these type of things that can all help to contribute towards your final conclusion. Then you should have around a full page of analysis and discussion just with writing. This will make up sort of the bulk of your analysis of your data, and it's quite important to really delve into that. Then again, this table format that I'm talking about for your evaluation, it's good to do this in a table format and include a description of the error that you made, the effect that this error had on your data, and potential improvements. It will save you space and time, and it will really help the marker go through it more quickly and more easily. And then, of course, cite your sources. So I was asked to come up with some possible IA ideas, since that's kind of the hardest thing that everyone has to come up with. They never know how to start. I didn't either. So this is some possible ones. You can go through these on your own as well later on. There are a lot also online, but these are just some possible ones that I've seen done or that I found as well. So the questions, these are the ones that have been previously sent to me that people had. So first of all, how do you know which test to use? For example, t-test, ANOVA, chi-square, correlation, those types of tests. So I actually found this video, which is linked on the bottom, that sort of goes into this, and I'm sure there are other resources as well, but just generally to go over it, you need to research what the tests are best used for and whether that applies to your data. So it needs to fit your data. There's no best test out there. It has to be appropriate for your data. So then you need to sort of consider what the purpose of your experiment is. Are you comparing something, or are you finding a relationship between things? And that will affect which test you choose, and then the type of data. So if you have categorical data, for example, like on the previous slide, it was like something about brands of toothpaste. So brands of toothpaste is categorical data, or continuous data would be like the height of a plant. And that also affects which tests you would use. So chi-squared is basically for comparison and categorical data only. T-test is for comparison and categorical and continuous data. And then correlation is for relationships, because you're of course finding the correlation between two things, and continuous data only. So hopefully that helps. And then this video down below, you can watch that as well. And then here, what do I do if my data does not support the null hypothesis? So this is kind of what I was saying before. All is not lost if your conclusion does not support or your data doesn't support your actual hypothesis. You just need to explain why, and especially go over this in the evaluation section. Sorry, there's a truck. So you need to then identify whatever trend is present, interpret that trend, and then try to scientifically explain it in your evaluation, and also compare it to the accepted scientific context. So if it's completely different, just say that and say why that could have happened. And then lastly, Rachel already kind of talked about this, but we're not sure if you're definitely not going to be just evaluated by your IAs, but it's not yet confirmed whether the IA is weighted more. So we have a couple minutes for questions. I'm going to stop sharing.
So I have a couple that got sent in, if you want to answer them, Ella. What's the biggest difference between a biology IA and biology EE? Or if you do an IA, you know, what is required more of you to do an EE?
Mm-hmm. Yeah, that's a really good question. I had the same question because I did a biology EE. And essentially it's the scale and the amount of time that you spend on it, from my understanding, and sort of the depth of it. So IA can be more simple, and it kind of has to be because it's shorter. Mm-hmm. If you look at like previous IA questions or like research questions and compare them to those of the EE, that will be good to give you sort of a better idea of how different the focus is. And for example, EEs can be more broad, they can be a bit more time consuming, and sort of more in that direction. That's also what I was told. And yeah, so your bio IA should not take as much time as if you're doing a bio EE.
Awesome. Fabulous. And another question, do I get higher grades for more complex experiments?
That depends what you mean by complex. If it's more original, if it's more, if you're demonstrating more initiative or more creativity, then you probably would get more points. It doesn't have to be, it actually shouldn't be much beyond the IB syllabus. So if it's really complex, if it's totally outside of the realm of what the IB is assessing, then that's not really the point. You're trying to apply the knowledge that you've learned in the IB. So try not to make it too complex. And I think, yeah, you need to be able to manage it and juggle it with all of your other IAs. But a certain level of complexity, is required. But I think that a lot of the points, as you saw in the criterion, are sort of mostly derived from the way that you present this. So if your method doesn't have to be that complicated. It just has to be an appropriate level. It cannot be one that you just found on the internet. And the way that you actually analyze this is really important.